Welcome to Lecture 14, where we will give an overview of the digestive system. We're now going to take a look at the characteristics of the mammalian digestive system. Mammals have specialized organs that are in close association with the digestive system to help process food. There are many accessory glands that support the digestive process. They do so by secreting digestive juices and enzymes into the alimentary canal. These accessory glands to the digestive system include the salivary glands, which can be seen in the diagram to the right, the pancreas, liver, as well as gallbladder. Food processing begins the moment that a food particle enters the oral cavity. Teeth will grind and help to mechanically break down the food. This helps to increase the surface area of the food for enzymes to act upon it. Salivary glands release saliva that contain a variety of components such as mucus to lubricate the food, buffers to help neutralize food, amylase to help break down starch particles, and as well as antimicrobial compounds. Once a food bolus is formed, the tongue will help to physically push it to the back of the oral cavity so that the food bolus can enter into the pharynx. As the larynx moves upwards, a structure called the epiglottis is pulled downwards, which blocks the trachea, or the opening to the respiratory tract. This will help to direct the food into the esophagus. Peristalsis will help to move the bolus down the esophagus and into the stomach. The forces of peristalsis are so strong that even if you are upside down, it will still push the food bolus into the stomach. Once the food passes down the esophagus, it will pass the cardiac sphincter and enter into the stomach. The stomach is important as it has many important functions. The stomach will secrete gastric juices to begin the process of chemical digestion. These gastric juices are comprised of hydrochloric acid to help denature proteins and pepsin to break apart peptide bonds. The stomach will also form a liquid suspension of the food called chyme. The stomach also is very muscular, which helps in the mechanical breakdown of food, and it can also help to store food before it enters into the intestine for absorption. This diagram demonstrates the epithelial lining and specialized cells that are in the stomach. Gastric glands line the entire lining of the stomach. These gastric glands have specialized parietal cells. These cells have the ability to actively pump hydrogen ions and passively allow chloride ions to enter into the lumen of the stomach. In the stomach, this will then combine to form hydrochloric acid. Chief cells release pepsinogen. This will eventually be converted to the active form pepsin whenever a low pH is encountered, such as the pH of the stomach, which resides around 2 when it is in active digestion mode. Mucus cells secrete mucus, and this is important to protect the epithelial lining of the stomach from digestion. The small intestine is important as it is the main site of macromolecule breakdown. Though the small intestine has a small diameter, it actually makes up the longest compartment of the digestive system. It consists of 3 main regions: the duodenum, jejunum, and ileum. The duodenum is the main site of digestion and occurs right after the stomach. As chyme from the stomach mixes with gastric juices from the pancreas, liver, gallbladder, and intestinal wall, much of the food particles entering into the alimentary canal will be fully digested. In the jejunum, this is the site where the most absorption occurs. As a result, there are many folds in the lining of the intestinal membrane of the jejunum, as well as the ileum. The ileum is the last site of absorption as food heads towards the large intestine. As chyme enters the small intestine, secretin is released. This causes the pancreas to release a substance called bicarbonate, as well as other enzymes such as trypsin and chymotrypsin. However, these enzymes are released in an inactive form. Bicarbonate is important as it can act as a buffer to help neutralize the acidic chyme as it enters into the intestinal area. Enzymes are also activated in the lumen of the intestine. The liver produces and stores bile in the gallbladder. Bile is comprised of salts that help to emulsify fats in the diet. They also contain various pigments from the breakdown of red blood cells. As digestion occurs in the duodenum, food will then move to the jejunum and ileum through peristalsis, or simultaneous contractions. This is where the bulk of the nutrients are going to be absorbed. Large folds in the intestinal wall called villi are lined with epithelial cells that have tiny microvilli on the apical surface, as seen in the diagram to the right. These folds, in both the villi and the microvilli, greatly increase the surface area for the absorption of nutrients. Also note that there are many arteries and as well as veins that are associated with the small intestine. This will help to ensure that these nutrients enter into the bloodstream to be distributed throughout the rest of the body. If we take a closer look of the lining of the small intestine, we will see that each villi has both a capillary network as well as a lacteal or lymph vessel. This will help to aid in the absorption as well as the distribution of nutrients into the body. Amino acids and sugars will enter into the bloodstream, while fats are transported through the lymphatic system. Capillaries bring this nutrient-rich blood from the intestines back to the liver via the hepatic portal vein. This allows the liver to regulate nutrient levels and also to detoxify any toxins from the blood before it goes back to the heart via the hepatic vein. After it reaches the heart, it will then be distributed throughout the rest of the body. Fats or triglycerides are broken down by a special enzyme called lipase. Lipase will break the fats or triglycerides into fatty acids and monoglycerides. These can then diffuse into the epithelial cells and are reformed and packaged into chylomicrons. These chylomicrons can then enter into the lymph system. Note that bile salts are important in breaking up the large fat globules from the diet. These triglycerides will then be broken down into the fatty acids as well as monoglycerides by the action of the lipase enzyme. These will then be built into chylomicrons. Once these chylomicrons are formed, they can then enter through the lacteal and enter into the lymph system. The large intestine is the main site of water resorption and is the location where feces is formed. There are three main regions to the large intestine. These include the colon, the cecum, and the rectum. The colon refers to the ascending, transverse, and descending colon regions. The cecum refers to a pouch at the junction site between the small and large intestine. It is believed that the cecum may assist in fermentation of plant materials in herbivores. In humans, the cecum has an appendix, and it is now believed that the appendix acts as a reservoir for symbiotic microbes to aid in the digestion and absorption of nutrients. The rectum stores feces prior to elimination from the body. As the digested food material passes through the large intestine, water is resorbed, and this aids in the formation of a solid waste called feces. If any infection or irritation occurs, this may disrupt the resorption of water. This would then lead to diarrhea. If feces moves too slowly, too much water may be resorbed, which may lead to constipation. Thus, it is important to have the proper consistency in the waste material as it is moving through the digestive system. We're now going to take a look at different adaptations that are present within the digestive system of vertebrates. We begin with understanding the role of dentition. Dentition, or the structure and function of the teeth in the oral cavity, can vary depending on the vertebrate's diet. For instance, carnivores typically have sharp and shearing teeth for killing and tearing off particles of the prey, while herbivores tend to have broad molars and premolars to aid in the breakdown of the plant material. There are also some generalizations with respect to the overall structure of the alimentary canal. For instance, herbivores typically have longer digestive tracts. This is because the plant material has plant cell walls, which make the material less digestive. As a result, by increasing the length of the digestive tube, you are also increasing the ability to extract these nutrients from this undigestible or hard to digest material. In addition, there may be mutualistic bacteria that also reside in the gut of various vertebrates. For instance, in the human intestine, there are mutualistic bacteria that produce important vitamins that are used to supplement our diet. Ruminant animals, like the cow, have multi-chambered stomachs. The stomach also houses mutualistic microbes to help aid in cellulose digestion. Ruminants will also typically regurgitate or chew their cud. This means that the food material will be recycled in between all of the chambers of the stomach. This will help to further break down the cellulose fibers and to also increase the microbial breakdown of the food particles. Since most organisms are not constantly feeding, the digestive system must be regulated, meaning that it must be activated when needed or shut down when not needed. The regulation of the digestive system typically involves two main systems, the enteric nervous system and the endocrine system. The enteric nervous system consists of groups of neurons that are responsible for controlling the digestive system. As an example, as soon as food enters into the mouth, this will activate reflexes that cause the production of saliva. This saliva will then be secreted from the salivary glands. The endocrine system, on the other hand, involves hormonal control. This can act on the digestive processes as well as the digestive organs. For instance, hormones that are released by the stomach and duodenum will help regulate the timed release of the digestive enzymes to optimize digestion and eventual absorption as the food material passes through the digestive system. Energy levels must also be consumed. If excess energy or more food is consumed than what the body needs, this excess energy can be stored as glycogen in the liver or muscle tissue, or it can be stored as fat deposits in adipose cells. This will ensure that the excess food is stored so that in periods where food becomes short or food is needed, these excess energy stores can then be used to provide the important nutrients for the body. However, if too few calories are consumed, these glycogen stores in the liver as well as the muscle can be broken down and converted to glucose. In addition, fat can also be metabolized as an energy supply. It is also important to maintain glucose levels in the body. In general, in humans, blood glucose levels should be within 70 to 110 milligrams per 100 mils. The regulation of glucose levels typically involves two main pancreatic hormones, insulin and glucagon. Insulin is the hormone that is released when blood glucose levels go too high. The release of insulin will stimulate the uptake of glucose from the blood, thus decreasing the blood glucose levels. Glucagon works the other way. When glucagon is released, blood glucose levels will rise. This is because glucagon is typically released if blood glucose levels fall below the desired range. This will stimulate the breakdown of glycogen, releasing the glucose into the bloodstream. Both hormones, both insulin and glucagon, target the liver. After a meal that is rich in carbs, insulin will stimulate the production of glycogen, while glucagon is released between meals in order to maintain glucose levels. In addition, the pancreas will produce hormones that help to regulate digestion, as well as producing bicarbonate ions and digestive enzymes. As a result, the pancreas is a unique organ that is part of both the endocrine system as well as digestive system. Please take a look at the diagram to the right to understand how glucose levels are maintained. We begin with a normal blood glucose range of 70 to 110 milligrams per 100 mils. If blood glucose levels increase, such as after a meal, insulin will then be released. The release or secretion of insulin into the bloodstream will then target the liver. It will then allow for the transport of glucose into body cells, and it'll cause this excess energy to be stored as glycogen. As a result, blood glucose levels will then fall. If we take the other scenario, if blood glucose levels tend to decrease, such as after a period of fasting, the secretion of glucagon by the alpha cells of the pancreas will then cause the breakdown of glycogen and subsequent release of glucose into the bloodstream. As a result, this will then increase the blood glucose levels back within a healthy range. We are now going to take a look at what occurs when there are some deficiencies within the digestive system. Diabetes mellitus results from insulin deficiency. This can be either in the inability to produce insulin or because of a decreased cellular response in the target tissues to insulin, typically resulting from faulty receptors on target tissues. As a result, blood glucose levels will rise. However, the cells are not able to utilize the glucose for energy. This means that fat typically becomes the main substrate for aerobic respiration pathways. However, the metabolites from fatty acid metabolism can lower the blood pH and can also disrupt sodium and potassium balance. In addition, high blood glucose levels can exceed the kidney's filtration capacity. As a result, glucose is typically detected in the urine of diabetic individuals. There are two main types of diabetes mellitus, type 1 and type 2. Type 1 is termed insulin dependent. This is because this type of diabetes typically results from the destruction of the insulin-producing beta cells in the pancreas. This is usually due to a hyperactive immune response in the individual. Type 1 diabetes usually appears in childhood, and treatment typically involves multiple injections of insulin. Type 2 diabetes is termed non-insulin dependent diabetes. This occurs when target cells fail to respond to insulin. As stated previously, it's usually due to defective insulin receptors. This type of diabetes usually appears later in life. This is also common in obese people and can also be due to genetic defects in either the insulin receptors or in some sort of mechanism in the typical response pathway. Treatment for type 2 diabetes typically involves proper diet, exercise, and may be modified with medication. And finally, with respect to regulating the various processes of the digestive system, it is also important to regulate appetite. In order to regulate appetite, there are many feedback circuits that are present. These feedback circuits involve neuronal networks that relay and integrate information from the digestive system to the brain. These circuits are very important in maintaining homeostasis with respect to the storage and metabolism of fat. These feedback circuits also regulate the secretion of long-term and short-term appetite hormones. Hormones such as leptin, insulin, and PYY or peptide YY act as negative influencers to help to suppress appetite, while ghrelin triggers the feeling of hunger and is considered to be a positive influencer. Take a look at the diagram to the left to understand the interaction of these particular hormones in controlling appetite. From this point, you should be able to understand the importance of a proper diet, and you should be able to trace a food bolus throughout the digestive system of a typical human.